Please. So, since our speaker has joined, so we are about to start the program with permission of Coordinator Journal Professor Dr. Muhammad Iqbal Chaudhry Saad. Sir.
أعوذ بالله من الشيطان الرجيم بسم الله الرحمن الرحيم الحمد لله رب العالمين الرحمن الرحيم مالك يوم الدين إياك نعبد وإياك نستعين اهدنا الصراط المستقيم صراط الذين أنعمت عليهم غير المغضوب عليهم ولا الضالين آمين. Let me begin by congratulating you all on this occasion of Holy Ramadan, a holy month for us in which our prayers are answered, and we try our best to be close to Allah subhanahu wa ta'ala. In this holy month of Ramadan, we are starting a very important activity of comstech. And this is an activity which is based on a core mission of sharing knowledge between the researchers and scientists in Muslim countries. I would not really take much of your time because the comstech would be introduced by my colleague, Ms. Khazima, and speaker would be introduced by Dr. Hina Siddiqui. But let me welcome Saad Omer, a very distinguished scientist from a top institution in US and perhaps in the world, and who is directly relevant to this field. And we are very fortunate to have him as the first speaker of comstech Distinguished Speaker series. And this series would actually go on for a long period of time. It's going to be timely, and this is going to be based on current needs of Muslim Ummah and humanity at large. So let me welcome Saad Omer. Saad, thank you very much for accepting our invitation. We are grateful. We know that it is early in the morning in US, perhaps immediately after the Sahar time. But I know this is a very blessed time also, and whatever you would do would, inshallah, increase your reward towards, inshallah, of everything, and your contribution towards Muslim Ummah would always be remembered. Thank you very much, Saad, for contributing.
Thank you. It's my pleasure to be here. Assalamu alaikum, everyone. I'll get right down to it in the interest of time, if that's okay with everyone. And I'll get started.
Okay, so if you allow, just brief introduction of comstech, because this is an organization which represents OIC. This is the OIC's Wing for Science and Technology. It is headquartered in Islamabad, and it is chaired by the president of ARTSA. So my colleague Hazima would actually introduce you to comstech.
Hello, assalamu alaikum everybody. This is Hazima Mozum from comstech. Thank you for joining us. Let me introduce comstech to you. Comstech is the Ministerial Standing Committee for Science and Technology. We operate in 57 countries. The core mandate of comstech is to enhance and strengthen scientific collaboration among the OIC states, with the ultimate aim is to build and nourish scientific culture in the Islamic countries, ultimately leading toward socio-economic development in these countries. Thank you very much again for joining us.
Hina, please.
Yes, sir. Hello, everyone. It's my pleasure to introduce all of you with today's speaker. Professor Dr. Saad B. Omer is the founding director of Yale Institute of Global Health, and he is a professor of medicine and epidemiology at the world famous Yale University, the School of Medicine and Public Health, USA. He has conducted studies in United States and Guatemala, Kenya, Uganda, Ethiopia, India, Pakistan, Bangladesh, South Africa, and other OIC states. Dr. Omer's research portfolio includes clinical trial to estimate efficacy and safety of maternal and infant influenza, pertussis, polio, measles, pneumococcal vaccine, and trials to evaluate drug regimen to reduce mother-to-child transmission of HIV. He has published over 280 papers in peer-reviewed journal and has mentored over 100 junior faculty in clinical and research postdoctoral fellow also. Over to you, Dr. Saad. Thank you so much for accepting our invitation.
Thank you. It's my pleasure. Assalamu alaikum, everyone, again. And before I start, I must acknowledge one of my professors from 20, 25 years ago, Dr. Pervez Iqbal. I think that's the same Pervez Iqbal that, if I'm not mistaken, from AKU. I can't see him properly, but it's really good to see at least one familiar face, at least one person who started me on my scientific journey. In addition to an overall salam, but a special assalamu alaikum to Dr. Pervez Iqbal. So I will start with a brief introduction of the field, and then I will go and talk about epidemiology, evidence-based policy, and then go into a little bit of a prospect of a vaccine or a series of vaccines, hopefully, for COVID-19. I'll try to keep some time, and I have attempted to keep some time from some questions, etc., from the audience as well. So where did this come from? Where did this virus come from? I think it's well known that it's a bat origin virus, but it did go through, is likely to have gone through an intermediate host. There are different speculations and some evidence around a couple of intermediate hosts, but I don't think we will, or at least for now, we have definitive data of what intermediate host it went through. That has implications, at least medium to long term. In the short term, we are focusing on controlling the disease. This is the current state of the outbreak. The US is now the epicenter of the outbreak. The virus grew from Wuhan. And a lot of us from the beginning have been saying that this tradition of naming viruses around populations has been there. So Spanish influenza, Ebola virus, etc. They're all named after places, but it's not the best way to do that, because you're already a population is suffering, and you don't want to stigmatize even more. So it's in more than 185 countries and territories, 3.7 million cases, and then over 25. Actually, now it's a little over 257,000 deaths. And so, just to step back, that this is probably was a single zoonotic event, single jump from an animal host to a human host in late November or early December. And now it has infected 3.7 million cases throughout the world, 3.7 million people around the world. So it is a humbling thought, but it also tells us how fragile our systems are. One of the things I would like to, there's a little bit of a formatting issue, I think, as I present, but bear with me. We have known about viruses for a while, and one way we measure the transmissibility is called the R0 or R0, the basic reproduction number. And it is a simplified conceptualization is that how many, in an immune naive population, how many secondary cases of a virus are there when a single case is introduced. So what is on average transmission to secondary cases in the context of a single introduction? Sorry, there's a few things in the chat. I thought I just wanted to make sure that they're not addressed to me and you guys can hear me properly. But yeah, coming back to this, measles is one of the more infectious common diseases we know. It has an R0 of 12 to 15. Ebola had 2, SARS had 3. And we think that for COVID-19 or SARS-CoV-2, which is the virus that causes the disease or syndrome COVID-19, it's 2 to 3. And still we are working it out. When this virus was new, and starting with how to address this and some of the policy responses, when there was the first case reported in the United States, and there were no cases or very few cases reported in the OIC countries, if any, I wrote this op-ed, this is in the New York Times, and I warned of the public, the US government, and other governments of how to respond to this outbreak. So remember, this is just 1 or 2 cases, but those of us who work on infectious diseases more directly were increasingly concerned and knew that this is going to go much farther than it was right now at that point. So I warned against three things. First, the responses should be led by scientists. And I'm bringing it up here is that a lot of people in this group are sought for in terms of their advice and their expertise by various policymaking entities. And the reason why I emphasize that for the US, and that applies, the lesson applies to others as well, is that we knew even at that time that this virus is evolving, and we were learning about this, and we continue to learn about this every day, every week. And so if you have a gap between the ability to assimilate emerging information and policymaking, you make bad decisions. So therefore, if you empower the scientists to lead a response or to be very close to policymaking, you lessen that gap and you make better decisions. The other thing was no false assurance. Communications researchers have told us that in these kinds of public health emergencies, in this kind of especially outbreaks and pandemics, etc., if you have a situation where you initially provide false assurance, in the long run, in the short run, it may reassure people and decrease population anxiety, but in the long run, it increases the likelihood of low trust in government and public health authorities. The last thing is we knew that we had to guard for misinformation in terms of scientific and public misinformation. And these warnings have proven to be unfortunately prescient in the US and in many countries, including many OIC countries. So earlier on, so it was a reasonable thing to do to go into extreme social distancing measures as the outbreaks was expanding and it was entering the exponential phase. But even from the very beginning, even from late March, those of us who said that it was okay to increase social distancing were concerned about. So this came out in early April. It was my plan for opening up was submitted in late March. It came out in early April, and what it had some features. Sorry, there's some feedback. I think someone has two sources of sound open, so I'm hearing a little bit of feedback. And so this came out in the Journal of American Medical Association. It had some features are now included in several plans that are out there to to safely open up and ease these restrictions. Because the idea of extreme social distancing, whether you call it lockdown, whether you call it shelter in place, is to provide some time for certain policies, and these are the policies for which you buy time. First of all, you can't have shortcuts around extensive testing and contact tracing. So you trace contacts, you isolate those who are test positive, quarantine the ones who are asymptomatic. So just to give you an idea, just to make sure that we're on the same page, quarantining means asymptomatic or people. Isolation means those with symptoms or who are test positive. And the other area is that in China, people don't realize that 64% of clusters were familial clusters, so in families and extended families. So the idea is to reduce household transmission and to focus on traditional treatment options that are useful in not just curing the disease, but even reducing time in ICU by 20 to 30% would have been helpful. So that was part of it. Now there are a lot of other plans, but this is the way to safely open up societies. And when a lot of countries are not all, unfortunately, are following this kind of an approach. And obviously other entities also came up with similar plans. So what happened when we put in place, we meaning the world put in place these social distancing measures that I'm sure all of you are aware of, where there were lockdowns or other kinds of social distancing measures. We did a very quick analysis in late March of reduction in mobility. So there is an app called City Mapper. that looks at mobility of its users, and they had data from 41 cities. Unfortunately, not um, uh, it didn't have a huge representation of Islamic countries, but overall we saw that we had a couple of interesting findings in terms of when we looked at mobility data. There was a decrease in mobility even without these lockdowns, etc., at a rate of 3.4% per day in March when these lockdowns were being put in place. However, the first thing is a lot of it was anticipatory. And then the second thing was that mobility after social distancing policies or emergency orders or lockdown orders actually did go up. So there was a combination of both voluntary and government-imposed restriction in mobility. So we did these similar analysis in the US, but using cell phone data by county. And we found was if 0 is the day of emergency declaration, and if you go backwards from there, so this is on the y-axis is median time at home. So the median time at home was decreasing because the weather was getting better, but it saw a jump, whether it was a rural county or an urban county or a metropolitan county or overall in time at home, minutes spent at home. And so that's encouraging, but again, it started going up, pretty in anticipation, but there was an impact of these orders. This is, comparison of countries in South Asia in terms of total confirmed COVID-19 deaths per million. So this is accounted for populations, and this is from a couple of days ago. And this is from South Asia, but so that there's less clutter. But a lot of other countries are showing similar patterns. And one of the countries that is not labeled, one of the arrows is Bangladesh. So this is Pakistan, Bangladesh, India, and Sri Lanka, and then compared to the US. So what it shows is that at least for these countries, the outbreak in terms of deaths, because cases are more vulnerable to underreporting because of testing, deaths are slightly more reliable. A few things that in South Asia, the outbreak is slower than the US, but it is taking off. The rate of change is there. The doubling time is approximately 7 days, and they're clustered together. So they're not out of the wood, but there is hope that at least at this point, some of the interventions seem to be working because compared to the rest of the world, where they were at the stage of the outbreak, there was these restrictions were imposed rather quickly. And so that gives them space to try to now open up with a little bit more safety and a little bit of, by imposing some efforts that will help these countries ease up the restrictions, because restrictions themselves have their own cost. So now switching to vaccines, since it's a broad group of people, I'll sort of introduce the concept. Vaccines are a preparation of killed microorganisms, living attenuated organisms, or living fully virulent organisms that are administered to produce or artificially increase immunity to a particular disease. So that's the definition I like more than some of the others. So essentially any substance, be it nanoparticles or killed pathogens, etc., that increase the immunity or induce the immunity in human hosts or animal hosts if you're talking about animal vaccines. So I will go through various types of vaccines, and then I'll come to generally various relevant types of vaccines, because there is a lot of work going on parallel technologies. So it's relevant to go through those a little bit, albeit rather quickly. So the most simplest concept, one of the more simpler concepts is this of live attenuated vaccines. You basically take a microbe, you weaken it by passing through, for example, cereal cultures, and reduce the disease potential by inducing mutations in that organism. And ideally, sometimes you have a non-replicating virus. Sorry, there is some feedback. So if people who have two sources of sound, if you could switch off at least one of them. Thank you. And so it is replicating, the ideal live attenuated vaccine replicates in the body. And this is better for viral pathogens because bacterial genomes are more complex. So there are several vaccines that come into this category: measles, mumps, and rubella vaccines, and oral polio vaccines, and even BCG. The way it works, as an example, is that the pathogenic virus is isolated from a patient and grown in human cultured cells. And then you take these culture cells and infect monkey cells, and there could be other cultures as well. And then the virus acquires many mutations that allow it to grow well in these monkey cells. And then as these mutations are induced, the potential for causing disease in human cells is attenuated. So that's the attenuation part. And so you take advantage of the fact that it's a live virus to induce stronger immunity, but you sort of neuter the disease potential. Then you have the killed or inactivated vaccines. A microbe is taken, killed by chemicals, radiation, or heat, suitable for bacteria because they have more complex genome. Weaker immune responses are induced through this approach, and therefore often require boosters. Then you have subunit vaccines. What you take is that you do the same thing. You take a microbe, and then you split it up, you disrupt it and purify it to take a subunit of that induce immunity, the relevant part of it. It has better safety and reactogenicity. Reactogenicity is immediate reactions you get after vaccination and may or may not have long-term sequelae. But so since you are just taking selective parts of it, often proteins, you have better safety and reactogenicity profile than the whole cell vaccine. Then you take these, in certain kinds of vaccines, you have the sugar molecules. Molecules are used for vaccination, but they don't induce a robust cell-mediated response, especially in children. So what you do is you take the sugar. I'm sorry, is there someone saying something?
लाइट खोल लिया करो यहाँ से
Okay, so I'll continue.
झाड़ू पोछे लगाते हुए लाइट पंखा खोल लिया करो फिर जाते हुए बंद कर दिया करो जब हो जाए.
So you have you culture, disrupt, you purify, and then you conjugate these sugar molecules with proteins, and you create a conjugate vaccine where you take advantage of a protein-based response, but the antigenicity of the polysaccharide part of the antigen. And it is particularly useful for young infants. And then there are these toxoid vaccines. You take the pathogen, look at this, you have this site related genicity and then epitopes that induce immune response. You do chemical modification and convert it to a relatively harmful toxoid. And this is for microbes that secrete toxins, etc. Then you go to a little bit more sophisticated technology, although recombinant vaccines have been with us for a while. The basic concept is that you extract the RNA, the DNA or RNA, and you insert it in a vector, such as yeast or bacteria. It could be other vectors. And then since you have inserted that genetic material, these entities produce the antigens. And these antigens are produced, and you get rid of the host, like yeast, bacteria, or another sort of host, and you purify the antigen, and that becomes your vaccine. And as you can imagine, this gives you more flexibility to create a vaccine. And it gives you more agility to do that. And the fact that this process has been there for a while means that although initially there were high development costs, now the costs have come down. And then you have the recombinant vector vaccines, where you insert the DNA or RNA. This is an RNA virus that we are dealing with in terms of SARS-CoV-2. You insert it in a vector. The proteins, the antigens are expressed by that vector, for example, on the surface, and you use the whole entity as your recombinant vector vaccine. So again, you have flexibility in choosing antigens. It has a relatively higher development cost, but that has come down. And so this is a platform that is being considered substantially for these kinds of vaccines. So you have naked DNA or RNA vaccines, where you extract the DNA, and so this is for the DNA type vaccines, but you extract the DNA, introduce it to the body, and integrate into the cellular DNA, and then you have antigen expression. These are relatively inexpensive. And then you have reassortment vaccines. You tissue the culture cells infected with 2 different strains, human and non-human sometimes. You induce viral replications, and they combine for multiple combinations of gene segments are produced in the offspring, and you take the offspring with desired characteristics, and that's your vaccine. So this is one example. I'll just, in the interest of time, I'll sort of urge you to focus on the left panel. So for example, this is an example from some reassortment vaccine, where you have a donor strain and a vaccine strain. You co-infect, in this case, an egg, but it could be a culture. You get potential reassortants, and you screen the reassortant that is desirable, and you use that as the vaccine. Now there's a technology which is relatively new, has been used in cancer therapeutics, and that's being employed, including for COVID-19. What you do is that you take in vitro translation, you induce in vitro translation and production of messenger RNA. You formulate it either into a nanoparticle or something else, and you introduce it into the body. So this mRNA, messenger RNA-based vaccine, goes in to the muscle, goes through the ribosomal processes where you transcribe and produce viral proteins. These become viral antigens, or are presented either through MHC class 1 or class 2 molecule, and those kinds of presentation. And the fact, since you are sort of using an upstream mechanism to quickly encode the messages for your protein production, you induce both a good B and T cell response. But these are experimental vaccines and have never been used in large populations. So this is a summary of the kinds of vaccines. You start from the top, you take, and some of them I covered in more detail because they're relevant to COVID-19 development. So you take the pathogen and you attenuate it. It results in an attenuated vaccines. You take the genetic material from the pathogen, you use a vector and induce it, and you take 2 strains, you merge them together, you culture them together, and you become it results in a reassortment vaccine. You have an inactivation process of the pathogen. You get the kill vaccine. You inactivate, disrupt, purify. It becomes a subunit vaccine, and you conjugate it with protein polysaccharide. It becomes a conjugated vaccine. But these are the ones where you extract the DNA or RNA, insert it into yeast or bacteria, express the antigen, purify. It becomes a recombinant subunit vaccine. You use the naked RNA vaccine. You insert the DNA into a vector, and that becomes a recombinant vectored vaccine. And if you go through the in vitro translation and create a messenger RNA, so it depends. You extract the RNA or DNA depending on the virus. It results in a mRNA vaccine. And so a little bit about how various vaccines induce immunity, especially against protein antigens, because that's what you are focusing on. So just to start with, if you see an antigen, so here's this antigen that is seen by B cells, that results in extrafollicular activation. So what is follicular? What is a follicle? Essentially, in your lymph nodes, either central or regional lymph nodes, you have these germinal centers where there is a lot of activity happening of cell multiplications, and reactions that happen and processes that happen within these germinal centers are called follicular reactions, and those that happen outside are called extrafollicular reactions. And so therefore, if you start with an extrafollicular reaction, you have differentiation of these B cells into plasma cells. And these plasma cells result in, and if you recall, plasma cells are the cells that produce antibodies. They produce low affinity antibodies, so they are low quality antibodies that have low affinity for the pathogen, and the levels are lower than some of the other antibodies, and that's the process that happens outside the germinal center. But here's what happened when T helper cells get involved, and some of the innate immune system cells, like these dendritic cells, present these antigens to the T helper cells. That induces a response where antigen-specific B cells, with the help of these T helper cells, then some of these B cells migrate to the germinal centers, to the follicles. And these follicles are anchored by T follicular helper cells and some of the other cells. And there is multiplication, and what results in a germinal center reaction. As a result, you have clonal proliferation, meaning that it's an exponential expansion of these cells, and you switch from IgM to IgG, and even IgA or IgE. And the other process that happens here is affinity maturation. So with successive generation, if the reaction is happening here, this is a high quality response. This is what you want to induce. You have the maturation of the affinity of these antibodies to the pathogen or initially to the vaccine antigen, so that you produce high quality antibodies. And then this differentiation into plasma cells, secreting large amount of antigen specific antibodies, etc. Some of these plasma cells, most of them have a limited half-life and limited life. They get eliminated. Some of the plasma cells migrate to survival niches, mostly so they get protected because they migrate to bone marrow, and so they survive. And this is important, how these cells that survive are important in inducing vaccine responses. So this is what you see, a generic pattern. You have a primary antigen exposure through this extrafollicular response. Then the B cell proliferation. This is the first time when you are seeing an antigen, either through vaccination or through infection. You have B cell proliferation and differentiation into plasma cells in the germinal center. So this stage is the follicular response for primary exposure. But then when the initial exposure happens, there is a decline in antibody levels due to the short lifespan of these plasma cells, and then you return to the baseline level. However, either if there is a booster antigen exposure or a viral or a pathogen exposure, since you have already these cells surviving, there's immune memory, you have a rapid antibody increase due to reactivation of immune memory. You have short-lived plasma cells that maintain the peak. You have an initial decline of these due to elimination of these short-lived plasma cells. However, a slower decline because of the antibodies produced by plasma cells reaching these survival niches in the bone marrow. So this is very quickly how memory is developed. Remember, it's the same reaction, the same follicular reaction that we have focused on. You have T-dependent antigens, as we just discussed. You go to the germinal center reaction, same as before, but now we are focusing on a separate pathway that happens. A lot of them, as I said, produce antibodies right away, but a few of these B cells differentiate into memory B cells. So that's the beauty of it, that our immune system remembers. And these memory B stream also other parts of the immune system and other in spleen and in other lymph nodes. So you don't want an immune response only in that same lymph node that saw the initial antigen. You want it disseminated in the body. So these migrations happen, and then they get to these extrafollicular areas this time of spleens and lymph nodes. And what happens, remember that there was a quick response. The reason why secondary exposure leads to this quick response is because the same antigen comes in. There's already a B memory cell there that differentiates and proliferates into plasma cells very quickly. And since it remembers from the germinal center reaction, the secretion, the antigen, there's a secretion of large amounts of high affinity antibodies in the serum. So these are the basic concepts. As we focus a little bit more on the vaccine development stages. So what we do is, just to to bring everyone on the same page, when you develop a vaccine, the first step is to identify a pathogen and you isolate it. You understand the pathogenesis, you identify correlates of protection. This is often antibody levels or other immune markers that are associated with clinical protection. And often you do that with imperfect understanding, unfortunately, of the pathogenesis. Then you design the candidate. You have this empirical approach, where you trial and error, but there are these more rational approaches that use bioinformatics, etc., or approaches such as reverse vaccinology that is used for candidate design increasingly. That you, for example, look at the antigen, and that's one of the approaches used for a lot of these vaccines that are coming down the pike. You looked at the target, the spike protein characteristics, you identify the target and work backwards from there. You do animal testing, usually animal model development, that what animal models are likely to be associated with vaccine development, associated with mimicking the response in humans. You look at immunogenicity. So immunogenicity is the purely immune response, not necessarily clinical protection. And you look at safety in animals. And then you file, in the US, it's FDA, Food and Drug Administration. In Europe, it's EMEA. In other countries, these are national regulatory bodies, and you file an application, not for approval right away, but to tell them that you are starting these trials. You start with phase 1 trials where you have tens of people vaccinated. You focus on safety. These are first in human trials. Immunogenicity is also measured. Dose response curves are also measured. Then similarly, you go increasingly to phase 2 trials, which are larger. You look at not just safety, but also immunogenicity. And they're often called phase 2B trials. These are proof of concept trials that start going into clinical outcomes. But these clinical outcomes are not final outcomes. These are outcomes that are proxy of important clinical outcomes. So these are proof of concept trials. And then you go into phase 3 trials, which is the trials where you look at efficacy, which is protection against clinical response, and then continue to monitor safety. And then you file the application for approval to any regulatory agency, either FDA in the US or other regulatory agencies. And then you continue to do post-marketing safety surveillance and effectiveness assessment, the so-called phase 4 of a vaccine assessment. And that doesn't end there. The approval is different from recommendation. And there are different bodies. In the US, it's called the Advisory Committee on Immunization Practices. At WHO, it's the Strategic Advisory Group of Experts on Immunization, etc. And then the next step is it's the either insurance coverage in certain countries or the countries buying vaccine. And that is how you deploy. As you can imagine, there are a lot of stages of it. And in COVID-19, we need to be cognizant of it. So after describing this, I want to take you through what happens. Again, it's still there in the traditional paradigm. It takes multiple years. You start with target identification, as I mentioned. You go through phase 1, 2, 3 trials, and then licensures. And you have go, no-go decisions at each of these stages. Here, in order to speed up vaccine deployment in a pandemic, we had quick target identification. But as we were identifying target, we already started these trials, human trials, we skipped a lot of animal models, and we are already there in first in human trials, and then we are going to do efficacy trials that will be sometimes in some clinical development programs include right away moving to doing a combined phase 1 and 2 trial and having a fast regulatory pathway. While this is done, so remember we said that you would need manufacturing, etc., and deployment. Usually you wait for large-scale manufacturing to happen after licensure. Right now, there are investments happening in building up these factories right now. For example, Bill Gates and the Gates Foundation has already invested in factories, and they're already building factories focused on the main technologies that are out there that are being deployed for developing vaccines, so that there isn't a huge delay in development and assessment of these vaccines and large-scale manufacturing. And so therefore that is happening. One big concern is that countries, including OIC countries, are not used to, or many countries at least, are not used to vaccinating adults. And a lot of this vaccination will happen in adults, and we don't have a vaccine delivery system that is focused on adults. So this is an overview of the kinds of technologies that are being employed right now. And so this has been updated only slightly, but going from left to right in terms of the columns, you have DNA and RNA vaccines, and you have inactivated virus vaccines, you have live attenuated, you have a vector vaccine. Remember, we talked about vector vaccines, and some of them can be replicating, the others can be non-replicating, that meaning that they're not reproducing within the body. And then these are protein subunit candidates. And then there are a few candidates where the technology is uncertain because it's hard to get data quickly in terms of where they're coming from. Some of them are single dose, but not all of them. So that's a consideration. Often we miss people in subsequent doses. Some of them are based on existing licensed platforms, others are not. And the speed of developing the product varies by the product. So RNA and DNA vaccines can be identified relatively quickly, and these are the ones that were the first vaccines to go into human trials. But some of the other ones have also gone into human trials. For example, the Oxford vaccines, which is a vector-based vaccine. And so therefore, the current scale of trials and production is varies, etc. by the product, and there are entities involved ranging from academic groups to small biotechs to large companies. I think OIC as an entity should invest in vaccine development and deployment. Unfortunately, the OIC countries are underrepresented in the vaccine development pipeline, because this is something that is a cutting edge of scientific development as well as has public health implications. So as we prioritize investments in science, these kinds of entities that both lead to developing basic science infrastructure but have public health implications will have to be priorities for investments, because otherwise, look, the countries who create these vaccines have a little bit more control about deployment of these vaccines and access to these vaccines, although there are efforts being done to increase access to these vaccines. Now you have production issues in terms of, as I said, that thousands of vaccines are needed for trials, but we need immediate scaling up. And this scaling up, as I said, is being done in parallel, but it has to be done under specific requirements called good manufacturing practices. So I'll just pause here. I went through a bunch of stuff rather quickly, but I wanted to make sure that we had time for questions and answers. And if you want to follow along in terms of the developments around COVID-19, including vaccines, but otherwise as well, feel free to follow me on Twitter.
Hello.
Yeah. Hello.
Hello.
Yes, I
Hello.
can. Assalamu
Assalamu
alaikum.
alaikum. Assalamu alaikum, Dr. Saad.
Yes.
It's a great pleasure to hear from you. You have made all of us very proud, and we are happy to see you, although virtually. But it's nice to hear from you, although you are away from us. 6,000 miles away or so, but our hearts are very near. So thank you very much. You have made us very proud. There is one question I wanted to ask you, and that is the dilemma in this country. You have lived over here. You have seen the life in Karachi, and there is a debate going on whether to a complete lockdown for a longer period of time, or we should go for they call smart lockdown, only focusing on the areas where there is some identification of the cases. So please, since you are an internationally renowned epidemiologist, please give us your opinion that in a third world country like Pakistan, where masses are living in very close conditions, where there will be 8 people in one room and all that. So for how long this complete lockdown can work, and how the United States has decided to relax some of the restrictions, and despite the fact that there are still thousands of people dying every day.
So two things. First of all, in a lot of situations, in terms of technology and development of vaccines, United States is a good example. In terms of the national COVID-19 response, the US is not a good example. And the reason why I'm saying that, that there is a reason why the US has the highest number of cases and pretty substantial mortality in the US itself. So the national response has not been very effective. Some of the regional responses, for example, in New York, Connecticut, et cetera, have been, and California have successfully, and Washington state have successfully controlled those. So here's the lesson from that and other countries, and based on sort of science of epidemics. I think it was appropriate. So one of the things we know that outbreaks, the way to control the outbreaks is to be preemptive. If you're waiting for the cases, and this is what the US did, the federal government waited for the cases to rise, and they said, look, there are so few cases, why should we go in for these kinds of extreme social distancing measures? What they ended up having, a situation where the epidemic went in to an exponential phase. And they went into half-baked, ineffective measures like travel restrictions, which rarely work in Western countries, because there's so much, they restricted travel for China, for example, and now we have sequencing data that is coming out. We did this collaborative work led by Nate Gruba, my colleague Nate Gruba, and our paper is going to be coming out in Cell, where we show that even by early March, the virus was not coming from China in the US, at least most parts of the US, it was coming from Europe. So the focus was on those travel restrictions when the horse had left the barn, and they've waited to impose those restrictions, and the outbreak had entered the exponential phase. In several countries like Pakistan, and Bangladesh and other parts of the world, because governments react able to not as it was in only one of us who are talk about I started talking about a plan to open up literally the day after it was imposed in northeastern United States, because it was very apparent that it's not a tenable long-term situation. So what you do is you go in early, but you only do it to buy time. And you buy time for the following: ramping up testing and contact tracing, identifying your risk areas, and then easing up slowly so that you keep control on the outbreak while having an economic activity, etc. Other countries that are densely populated have shown, so Hong Kong is highly densely populated. Obviously, the circumstances are different. Parts of Korea are highly densely populated, but they were able to control the outbreak there. So the key is smart lockdown, but do it in, the opening up should happen now. So the, even in, province of Sindh, which was more aggressive and proactive, they're also talking about, and we are engaged with several entities, even within Pakistan, to help plan for smart lockdown, meaning easing off safely. And that's the key. You ease off it is time to ease off these restrictions, as it was time to go in early, but do it smartly, do it sector-wide, do it by testing people who are high risk of exposure, or not just high risk of exposure, but high risk of transmission, and so on and so forth.
Thank you.
Thank you very much
for being part of this program. And it was a great talk, a great learning for most of us. Regarding the vaccine development, as you highlighted, and the process that generally happens or should happen, there are so many steps that are being compromised, and we have gone into basically, quote unquote, almost like a marketing strategy straight away in order for massive development. And even before the development, we have gone into the Bill Gates Foundation and others are working on it. So as a scientist, purely from that point of view, it is a fearful thought process, isn't it? That, you're going through all that bit without going through the norms of practice as otherwise you would like to do.
So there are a couple of things. First of all, there are two things that have happened. It is remarkable speeds, but the speed is in steps that do not compromise safety and efficacy statement, safety and efficacy assessment. So the speed was in target identification. So the Chinese published the genome in January, as early as January, and that was used for early target identification. So that doesn't impact safety or efficacy directly, as long as you evaluate it properly. The speed is in production of the initial seed lots for trials. The speed is now on manufacturing. So manufacturing is okay to invest in right now, because that's a sort of a scale-up process. You develop that, you invest in that, and for example, it's okay. Bill Gates said, even if 6 out of 7 or 5 out of 6 factories go to waste, it is worth it because you gain several months by investing in it right now. In fact, it is safer to do manufacturing now because you have some more time than do it under a time pressure of 2-3 months after your vaccine is approved, etc. So you can ensure that the basic processes are similar. So even if, for example, if you're doing a vectored vaccine, irrespective of what the product is, there are certain processes that underlie those. So you do that, and you even use the candidate vaccine to say, okay, this is how we're going to scale up. So this is actually, they're expanding the timeline there compared to, let's say, an influenza pandemic. The other part, which is taking the same amount of time or similar amount of time, but being done concurrently, is the clinical evaluation. And a lot of us therefore insist that gain time through logistics in that part, but don't gain time for the natural process. For example, have the same sample sizes that you would have under normal circumstances, but enroll them in multiple sites so that is happened concurrently. So enrolling, if your sample size is 100 and you're enrolling at one site, it takes longer time. But if you're enrolling at 4 sites, it takes a quarter of that time to enroll them. but it's the same biological and clinical evaluation process. Don't compromise on the length of follow-up. For example, if the science warrants following up for 4 months after you vaccinate for efficacy and safety, take that full 4-month period, because that's really important to do that. So it depends how you speed up the process. The vaccine process has been more reassuring, frankly, than the treatment process. For example, the announcement from the White House in the US of hydroxychloroquine was not very wise. The trials hadn't been done there, etc. The vaccine process is better managed, actually much better managed, because it's been driven by science, and it's been driven by sort of an international process more so than a country-by-country process. So far, the speed is gained through logistical steps and preclinical steps, rather than taking shortcuts in clinical development.
So the question basically still arises that, when you come to vaccine, we don't know which group you're going to vaccinate and how successful it's going to be. Where is that kind of data? We haven't got the age brackets, for instance, 50 to 70 and 70 to 90, 30 to 40. In that age groups, what is happening? What should be the dose? Those trials are still yet to be seen, and the results would identify. And is the IgG protective? For how long it's going to be protective? What are the titers that I need to have in order to stay protected for what length of time? So there's so much of missing data, in fact, in those aspects of clinical studies that, as a scientist, I would have thought that you would elaborate some of that. If you
That's
may,
a really
please.
good point. Frankly, this is exactly the kinds of studies we are doing right now. The only difference is we are doing it in parallel rather than sequentially. So correlate of protection studies, what kind of immune response is associated with clinical protection? For example, we have a health worker cohort, and we have a course. So one of the things we established earlier on here at Yale with various partners is we have a biorepository, a tissue biorepository, where we enrolled from day one, since our patients started enrolling, or within the first week, we have been enrolling under really hard circumstances. We have a really big outbreak happening. We have a tissue biorepository that has PBMCs, that has, obviously sera is easy. We have nasopharyngeal swabs. We have, stool specimens in a lot of them, saliva. We have bronchoalveolar lavage, precisely for that reason. And so now the work is moving to identifying correlates of protection. The difference is we're doing it in parallel because the cost of inaction is so high. The way the world is dealing with it is by doing these things in parallel. But I would be the first one to say, take a breather if there are any shortcuts being taken in terms of clinical evaluation, because that time will come once you have phase 1-2 trials, when you have looked at initial safety and immunogenicity, and then the time will come and you say, okay, no, this is the time to, again, look at it with the correlate of protection, with efficacy and that kind of stuff. In terms of the age groups, that's the other thing that we are talking about. So we know in terms of what are the priority groups from a clinical and public health perspective. We know that the priority group will be, the top 5 are likely to be the following. The elderly, the healthcare workers who are engaged in high-risk procedures. For example, there's a higher risk when you're intubating, much higher risk as a healthcare worker if you are intubating someone versus if you're just taking their history. And so high-risk workers, then it will be people with comorbid conditions, irrespective of age. Then will be other healthcare workers who are not high risk, and then you go down the list. So that's the and that way we can plan. And one of the things what you pointed out will have to happen is some evaluation in those target groups. So health work, I know that there will be studies in healthcare workers for phase 3, but I would also want to see a study in the elderly specifically, because influenza, the responses are lower in the elderly compared to the other age groups.
Thank you so much. Thanks for your answers. Brilliant.
Ladies and gentlemen, in the interest of time, we can have one quick question if there is any.
Yes, please. A lot of countries right now, they are trying to reopen and go back to normal life, while still there is no potential therapies, no precise vaccine. And it seems like the precise vaccine is still like such a feat like a pass away. So what do you think? Is it wise to reopen and go back to normal life in these countries?
It depends on the situation. If your rates are high and still going up, you at least have to do partial lockdowns. So you can go into a situation where you're opening up parts of your country based on the local risk, but it would be dangerous to open up the whole country if your rates are still going up. If they're stabilized or going down, if it's a controlled outbreak situation, you have more flexibility at the national level, but you should do that carefully. And we need to, as societies, unfortunately, need to live, even when you open up in situations where you still have social distancing measures, still have measures for example, at least 6 feet distances. We need to recognize as a society that there will be congregations that we will have to advocate that it's not in the public health interest for them to happen. Large gatherings that are potential for super spreader events, like weddings, like sports events, especially indoor sports events, and unfortunately, large religious congregations, large salah prayers will have to be reduced. There's a reason, irrespective of your perspective, there's a reason where a lot of Muslim countries have reduced that. And as scientists who have public's interest, and that is our religious responsibility as well, we have to be advocate for rational policy there. So as you open up safely. So opening up will have to happen. Because otherwise you'll end up inducing more mortality due to, let's say, malnutrition and other things that are long-term consequences. So both things can be true and are true, that the initial lockdown was essential to buy time, to get things under control. But how you open up depends on the local city. And the last thing to get to normal, I don't see a situation unless you have hundreds and thousands of very large scale mortality, and there's so many people have gotten infected that there's no point in doing anything, but that would be a catastrophe of unimaginable proportion. Unless you have a vaccine, I think we should all expect, even when we opening up would mean that large scale events won't happen for a while until we have the vaccine, that there will be social distancing measures like at supermarkets, like at grocery stores, like at other places where we reduce the probability of transmission, and so on and so forth.
All right, thank you very much.
My pleasure.
Thank you so much. With this, I would like to thank our eminent speaker, Professor Dr. Saad Omer. Thank you, sir, for such an informative lecture and discussion session. Thank you so much. And I would also like to thank all the audience from various countries like Egypt, Azerbaijan, Bangladesh, Nigeria, Saudi Arabia, and many other countries. There are many participants from different cities of Pakistan. I also thank all of them. And I also would like to thank my colleague at comstech. And kindly note that the recording of this lecture will be uploaded on the comstech website for your reference, and if you would like to go back to the recording. Once again, thank you so much, everyone. Goodbye.
My pleasure.
Bye. Bye.
Thank you, ma'am.